Okay, hello everyone. Let me welcome you at the beginning of this course. Let me welcome the new generation of our students, medical and dentistry students of the Faculty of Medicine in Novi Sad, Serbia. This is one of the available online platforms where I'm gonna provide you the lectures which are part of the Biochem course for the students of dentistry and medicine at our faculty. And I think that it's going to be one of the useful tools for online and e-learning and preparation of your biochemistry course. On this slide you can notice my contacts. You can see my contacts, so please feel free to contact me at any time if you have any questions or if you need any kind of additional explanations. So my email is available, which is written here on the slide, and also I'm pretty active on social networks in Instagram, Facebook, Messenger, and so on. And of course my YouTube channel is always there to support you and help you in studying and preparing these biochemistry topics for our exam. So let's start with the first chapter, and that is the chapter on amino acids. So we are slowly starting to evaluate and to talk about different types of biomolecules, about their structure and properties, and we're going to start with proteins and amino acids as building bricks of protein molecules. So in the chapter on introduction on biochemistry, you discussed about the formation of biomolecules in ancient times, and you said that like 4 billion years ago the first biomolecules were synthesized from very simple compounds which were available at that time in those primary oceans and primary atmosphere on Earth. And if you remember that we stated that amino acids were actually first organic molecules to be synthesized from very simple molecules like methane, hydrogen, nitrogen, and so on and so on. Later on, upon the polymerization of amino acids, the first biomolecule was synthesized, and that biomolecule was protein, or those biomolecules were proteins. Also, we can freely say that proteins are the most important biomolecules for the structure and function of living organisms. Actually, even the name protein originates from the Greek name proteos, which means the first or the one which is in the first place. So basically, proteins are polymers of amino acids. That is a very well-known fact. And those amino acids are attached to each other by peptide bond, and we're gonna elucidate both amino acids and peptide bonds in the upcoming chapters, this and the following video. But let's start with amino acids first. There are 20 proteinogenic amino acids, and those 20 types of amino acids are actually components of human proteins. And we're gonna start with a story on structure of those 20 proteinogenic amino acids, on classification of those, and their physicochemical properties. But apart from these 20 proteinogenic amino acids, there are some amino acids which are additionally modified after incorporation in the protein molecule, and we're gonna say a couple of words about those too. Also, in our body, in human organism, there are some amino acids which are not involved in the formation of proteins, and those are marked as non-protein amino acids, but nevertheless they exhibit very important biological functions. So some of those amino acids can act as hormones, biogen amines, neurotransmitters, etc., etc. We're going to mention some of the examples, and a lot of those amino acids will be topics in the upcoming chapters of our biochem course. So, one can think, okay, it's not too much. It's only 20 amino acids, so proteins are very simple molecules, and it's not a big deal if you have only 20 possible molecules to incorporate in the structure of a protein. But if you take into account that an average protein molecule contains approximately several hundreds to several thousands amino acids in the structure with an average molecular weight reaching 10 to 5 daltons and above, and if you try to mathematically calculate the number of possible combinations of those 20 amino acids, if you have the total number of several hundreds or thousands, in the end you're going to get a huge number. So you're going to get an extremely large number of possible combinations. But let's start first with the structure of amino acids. So here on the slide you can notice the general structure of one amino acid, and you can notice that there are two functional groups in each molecule of amino acid, and let's summarize them. The first functional group is amino group, NH2 group. Let me remind you a little bit on your knowledge from the organic chemistry. And the second functional group is carboxylic group, COOH group, and here it is. What can we notice from this structure? We can notice that both functional groups are attached to the same carbon atom, which is, if you know from your organic chemistry course, is marked as alpha carbon atom because it is the first carbon atom next to the or adjacent to the carboxyl group. So all human amino acids are marked as alpha amino acids, and both functional groups are attached to that same alpha carbon atom. Let's move on. If you take a look again at the structure of one amino acid, you can notice that amino group, which is written in this figure, is written on the left side, and if it's so, that amino acid is marked as L-amino acid. But there is another possibility, and that other possibility, second possibility, is that amino group is positioned on the right side. In that case, we have the form which is marked as D-amino acid. So which of those, or both, are involved in the structure of human proteins? We can say, and we know, that only L amino acids are found in human proteins, and D amino acids are not found in human proteins, and moreover, D amino acids may exhibit toxic properties too. So let's conclude. If you take a look at this structure, and if you take in account these conclusions that we have made, we can say that all human amino acids are actually alpha L amino acids. There is one exception, because there are always some exceptions in biochemistry, and you're gonna get used to it actually. That is an amino acid proline. What is the exception? The exception is that it has imino group instead of amino group. And what is left here? The third bond of the alpha carbon atom is reserved for hydrogen. So this upper part of the structure is common and same for all amino acids, but they actually differ in the structure of the fourth atomic group attached to the alpha carbon atom, which is marked with a capital letter L. So amino acids actually differ according to the R residue, and this R residue is marked as side chain group. So, if we would like to somehow classify those 20 proteinogenic amino acids and thus explain their structural properties, we can do it in numerous ways. So if you're using different textbooks, or if you're using some other internet sources regarding this topic, you may find that different authors are using different classifications to classify amino acids into certain groups. But I have chosen to present you two ways to classify amino acids, which I have actually found to be the most appropriate for beginners, for those people and students and candidates which are not that familiar with biochemistry and chemistry. So I hope that these two ways to classify amino acids and simultaneously explain some structural characteristics are going to be easy for all of you. So this is the classification according to the structure of the side chain, and the other classification is according to the properties of the side chain. So let's start with the first classification, which actually classifies those 20 amino acids in 8 groups. So the group number 1 are amino acids with aliphatic nonpolar side chains, and there are 5 representatives or 5 amino acids belonging to this group: glycine, alanine, valine, leucine, and isoleucine. So you can notice that they actually have hydrocarbon chain in the side chain. Also, you can notice here in brackets that abbreviations are used to mark amino acids in simple way, which is especially useful when listing the sequence of amino acids in peptides and proteins. And the most common abbreviation method is to mark each amino acid with 3 letters, and usually those 3 letters are the first 3 letters from their name. So glycine is marked as GLY, alanine as ALA, valine as VAL, and so on and so on. Again, if you're using some other textbooks or sources for preparation of this chapter, you may notice that there is one other way to mark amino acids, and that is, or to abbreviate amino acids, and that is with 1 capital letter, which is not necessarily the first letter of the name of the amino acid. And this kind of marking is more complicated, and I think at this level of the biochem course it's absolutely unnecessarily for you. Also, of course, you can notice from this first group, and that is the same for all other groups of amino acids, that we usually use their trivial names instead of UPAC names. And if you are interested, you can easily Google the UPAC names for these amino acids. But again, I think that it's not that important to memorize those chemical names, UPAC names, because literally everyone is actually using those common names for amino acids. So if we take a look at this group and the representatives, we can see that the amino acid glycine is very simple. It has only hydrogen atom in the side chain. And the things are getting slightly more complicated, approaching 2 other representatives. So alanine has methyl group in the side chain, valine isopropyl group, and so on and so on. But the common characteristic is that they have hydrocarbon chain in the side chain, thus having an emphasized aliphatic non-polar properties. So the group number 2 are amino acids containing aromatic ring in the side chain, and we have 3 representatives: phenylalanine, tyrosine, and tryptophan. All of the 3 containing benzene ring, so aromatic ring in the structure. In phenylalanine, it's pretty simple, so only benzene ring attached in the side chain. Tyrosine with the OH group added, but despite the fact that it's OH group added, this amino acid is considered to be non-polar, so polarity of this OH group is not that pronounced. And tryptophan with specific indole ring, so the system of 2 condensed ring. If you remember our organic chemistry course, we discussed about heterocycles, and we mentioned indole ring, which is specific for tryptophan. These aromatic amino acids, they exhibit one very interesting property of the benzene ring, which is to absorb the UV light at 280 nanometers, which is used in laboratories for quantification of not only amino acids but proteins as well. Third group of amino acids are amino acids containing hydroxyl group in the side chain, OH group in the side chain, and we have 2 representatives, serine and threonine. Fourth group are amino acids containing carboxylic, additional carboxylic group in the side chain. So in the end, they have 2 carboxylic group in the structure. We have 2 representatives, 2 amino acids belonging to this group, aspartic acid and glutamic acid. The fifth group are amino acids containing an amide group in the side chain, and we have again 2 amino acids belonging to this group, asparagine and glutamine, and notice the structural similarity to the previous group. So comparing to aspartic acid, asparagine only has amide group instead of carboxyl group, and the situation is analogous between glutamic acid and glutamine. Sixth group of amino acids are amino acids containing 1 additional amino group, at least 1 additional amino group in the side chain, thus exhibiting basic properties. So we have 3 amino acids in this group: lysine, arginine, and histidine. Lysine with amino group and hydrocarbon chain, arginine with very specific guanidino group, and histidine with imidazole ring, if you remember our story on heterocycles. Seventh group, and we're almost done, are amino acids containing sulfur in the structure, in the side chain, and again 2 amino acids in this group: cysteine with SH thiol group, which is a polar group, and methionine, where this thiol group is methylated, if you can see. And finally, in the 8th group, we have our exception, and that is amino acid, proline. So notice that instead of amino group, we have amino group in the molecule of proline. So these are 20 proteinogenic amino acids. But apart from this classification according to the structural characteristics of the side chain, we can classify them according to the properties of the side chain. And some of the properties I have already mentioned. So according to this, 20 amino acids are divided in 2 large groups. The first group are non-polar or hydrophobic amino acids, and the other group are polar or hydrophilic amino acids. So in the group of non-polar amino acids, we can find those containing non-polar hydrocarbons chain, or benzene non-polar aromatic ring, or our amino acids. So alanine, valine, leucine, isoleucine, phenylalanine, tyrosine, tryptophan, and proline are amino acids, which are non-polar and hydrophobic. All other amino acids are considered to be polar or hydrophilic. So let's make a list and let's make some subclassifications of this group. So we have glycine, serine, threonine, cysteine. Serine, threonine, you know, they contain polar OH group in the structure. Cysteine contains polar thiol SH group. And glycine is actually a kind of controversial. So again, if you're using some other textbooks, additionally, some authors claim that glycine belongs to the group of non-polar amino acids. But if you take a look at the structure of glycine, you notice that there is only one hydrogen atom in the side chain. So actually the properties of the alpha carbon amino and carboxylic group predominates. So you will see in a couple of minutes that actually those two groups are charged, protonated or dissociated. So actually glycine might exhibit properties which are similar to some dipole molecules with asymmetrical distribution of the charge, electrical charge, like in the molecule of water. So in that case, and because it's a very small molecule, it can be considered a dipole and thus being a polar or hydrophilic amino acid. But let me point out here that literally no one is going to make a big deal, or it's not going to make an issue if you classify your glycine molecule to be non-polar or polar. I think that each and every one of us is going to accept both answers. So also in the group of polar amino acid, we have acidic amino acids. Those are amino acids containing additional carboxyl group in the side chain. So they are also marked as monoamino dicarboxylic acids, and we have two of them, aspartic acid and glutamic acid. Then we have neutral amino acids, which are also polar and hydrophilic. Those are asparagine and glutamine, our amino acids containing an amide group in the side chain, and of course basic amino acids, which contain additional amino group in the side chain. And if you remember, we have three basic amino acids, which are lysine, arginine, and histidine. And sometimes they're also marked as diamino monocarboxylic because of two amino groups in the structure. But apart from this classification, these two ways to classify, one more issue is important to state here, is that these 20 amino acids are also different, having in mind their capability to be synthesized by our enzymes and by human cells. And approximately half of them, 10 of them, cannot be synthesized by human cells because we don't have any molecular mechanisms available for the biosynthesis of those. So those amino acids have to be provided through diet. So dietary intake is required for these amino acids. And whenever you have such a situation, and we're going to have it with other biomolecules as well, those molecules are marked as essential. So essential amino acids are those which cannot be synthesized by human cells due to the lack of proper biosynthetic mechanisms and enzymes. So dietary intake is required. 10 amino acids are considered to be essential, and those are listed below: valine, leucine, isoleucine, phenylalanine, tryptophan, histidine, lysine, arginine, methionine, and threonine. Let's move on. I have mentioned that there are some amino acids which are found in our proteins, but those amino acids are chemically modified after the completion of proteosynthesis, or we are using the term they are formed by post-translational modifications, having in mind that translation is the term for protein synthesis. Of course, there are a lot of examples of these, but I have chosen to present you just a couple of common examples. So in collagen fibers, and medical students are going to deal with connective tissue and collagens and keratins and elastin more than dentistry student, but 2 amino acids are found in collagen fibers. Those are 4-hydroxyproline and 5-hydroxylysin. So you can notice that OH group, which is green shaded, is actually additionally attached to the primary molecule of proline and lysin during the biosynthesis of collagen fibers. In myosin fibers, you have 2 methylated amino acids, N-methylysin and N-methylhistidine. In prothrombin, which is inactive form of thrombin, which is involved in blood coagulation, you have gamma carboxyglutamic acid, so 1 additional carboxyl group attached in the side chain, and we have 2 rather complex molecules of desmosine and isodesmosine in elastin fibers. So medical students will experience the story on collagen and elastin later on. So I'm gonna stop now. I'm not gonna force you at this specific moment with the structure of elastin. Also, I have mentioned that there are some non-protein amino acids which might exhibit very important biological functions. Beta-alanine, for example, it's the degradation product of pyrimidine nucleotides. Citrulline and ornithine are involved in detoxification of ammonia, thus forming a urea molecule in liver. Gamma-aminobutyric acid, or GABA, is neurotransmitter, I think very famous molecule. I haven't mentioned here, but for example, some hormones are derivatives of amino acid tyrosine, so they're not involved in protein structure, but they have very, very important biological functions. So, for example, dopamine, adrenaline, noradrenaline, thyroid hormones, they all they're all derivatives of amino acid tyrosine. Let's move on. Slightly something about amino acid properties. So the first one is optical activity. So if we get back to the structure of the amino acid, we can notice that there is at least one, maybe there are more, but at least one chiral carbon atom. It's asymmetrical, meaning that 4 different atomic groups are attached to that carbon atom. If that is the situation, so if a molecule has an asymmetrical or a chiral carbon atom, it's considered to be optically active, thus rotating the polarization axis of linearly polarized light to left or right, depending on the structure of the molecule. If a molecule is optically active, I'm going to remind you that there are 2 possible forms or shapes of the molecule. They're called enantiomers or optical isomers, and there are 2 types, D and L. If you remember, we have already noticed that in humans only L enantiomers or L amino acids are found. There is one exception, and that is amino acid glycine. This one is not optically active because if you remember, glycine has only 1 hydrogen atom at the position number 4 in the side chain, and actually then the alpha carbon atom actually has 2 identical groups, 2 H's, so it cannot be optically active. Another very important property of amino acids is acid-base property. So we know that each amino acid has 2 groups, carboxyl group and amino group. We know that carboxyl group is a carrier of acidic properties of an organic molecule, so we can conclude that amino acid may act as an acid. Thus, its carboxyl group can dissociate in an aqueous solution to carboxylate anion and proton or hydronium ion. On the other side, we know that amino group is a carrier of a basic properties of organic molecules. So, amino acid may act also as a base. Thus, in an aqueous medium, amino group may accept proton, thus forming the protonated form, and hydroxide ions is released, giving the basic or alkaline reaction. So we can have both stories. So amino acids are at the same time acids and bases, so they can act both as acids or bases. So I'm going to remind you again on the chemistry a little bit. So when we have a molecule with such properties, we consider it an ampholyte. So amphoteric properties are specific for ampholytes, and amino acids actually are ampholytes. The next important property is the form in which amino acids are found in aqueous solutions. So in an aqueous solution at physiological pH, so it's kind of neutral medium, amino acids are actually found in dipolar or Zwitter ionic forms, meaning that carboxyl group is dissociated to carboxylate anion, which is negatively charged, and amino group is protonated to NH3 plus 4. But the change of the pH of the solution, I'm sorry, I forgot to translate this, but the pH change actually might change the form of amino acid in an aqueous solution. So in highly alkaline medium, upon the addition of hydroxide ions, this NH3 plus group is going to dissociate to NH2. So the overall structure and the overall charge of amino acid is actually negative, so amino acid becomes an anion. On contrary, if you're going towards a highly acidic medium, the addition of hydrogen ions is gonna additionally protonate carboxylate anion to COOH form. So the charge of the molecule of amino acid is going to be positive, so it's going to be in a cationic form. Another properties arising actually from this are buffering properties of amino acids. So you know what are buffers? Buffers are systems which can resist the addition of small quantity of a base of an acid with no significant changes in the pH. So the highest buffering capacity is actually noticed for amino acid histidine. I mean, generally, all amino acids are good buffers, but histidine is the best among them because of the presence of imidazole ring. And if you remember the structure of that imidazole ring, you have one more hydrogen on one nitrogen from the heterocycle, which is also capable of getting involved in the maintenance of the pH. And what is interesting here to mention, and you will repeat it a lot of times in the upcoming chapters, hemoglobin molecule contain a significant portion of histidine in the structure. Approximately 8 to 10% of amino acid content is histidine, which makes hemoglobin very important physiological buffer, and you're going to discuss this in details later on. And finally, to conclude, its isoelectric point of amino acid, that is the pH, at which amino acid actually loses its electrical charge and it becomes zero charged. It becomes a neutral molecule without any kind of mobility in electrical field. Because it becomes neutral, it is much less soluble in an aqueous solution with a tendency to precipitate. This isoelectric point, which is usually marked as P, capital letter I, is used for separation and isolation of both amino acids and proteins, and I'm gonna get back to the story on isoelectric point later on in the next video in a chapter of proteins and their physicochemical properties. So anyway, my dear students, my friends, my dear everyone, this was what I actually wanted to tell you regarding the amino acids, some of the basic information, but I think sufficient information for us to successfully understand this chapter. So until my next video on peptides and proteins, I'm gonna say goodbye for now and have a nice rest of the day. Enjoy biochemistry and see you very, very, very soon.